So what are all stages that the membrane must move through when transmitting a signal? I just said that. Uh, what is the concentration gradient of the membrane at resting potential? The concentration gradient is that it is more positive on the outside relative to the inside. And how are the signals able to travel along the membrane? And if you saw the diagram earlier, you can see that as one section of the membrane depolarizes, neighboring sections also depolarize, and that's essentially how it's able to move along the membrane.
Okay, and here's some tertiary stuff to the big picture behind transmission of a signal. This whole transmission of a signal is going to end up with a release of neurotransmitters. And these are the signals that are going to move on to the dendrites of the postsynaptic cell. And these can range anywhere from known ones to serotonin and acetylcholine to smaller neuropeptides and biogenic amines. And these are all going to provoke some type of cellular response that the body is trying to create.
Okay, so signal summation is essentially what it sounds like. It's a summation of two signals. You have spatial and temporal. In spatial signal summation, you have two signals which are in neighboring neurons, and they happen at the exact same moment, and as a result, the signals add up, creating a stronger one signal. And then you have temporal signal summation, which is where you have two signals. Essentially you have one signal traveling down the axon, and the signal behind it catches up to it. And as a result, the two signals add in the plainest of terms.
And last we have inhibitory postsynaptic potentials and excitatory postsynaptic potentials. And this is another example of how neurotransmitters affect the next neuron. Inhibitory postsynaptic potential is what it sounds like. The neurotransmitters are going to move the neighboring neuron away from reaching action potential so that there will be no next response. And excitatory postsynaptic potentials are going to do the opposite, and those neurotransmitters are going to excite the neuron that they're transmitting to, and they're going to bring that neuron closer to having a change in membrane potential that will incite action potential.
Okay, this is our last bit of questions. Alright, for the first one, we have a scenario. Somebody is being chased by a wild boar and breaks their leg after falling, but gets back up and is able to continue running. What type of neuropeptide is most likely at play? The answer to this would be an endorphin. Endorphins essentially block pain receptors, and they're a type of neurotransmitter, so they dock with the receptor and prevent pain signals from being transmitted. Where's the one place that the vertebrate neurons do not release acetylcholine as an excitatory transmitter? And this is the heart. The reason for this, one could guess, is that you don't want a variety of signals going into the heart because it's extremely rhythmic and needs to maintain its pace. Explain the difference between spatial and temporal signal summation. I explained those earlier, but for a brief recap, spatial summation is when two different neurons transmit a signal at the exact same time in the ad, and temporal is when the same neuron transmits two signals and they add together. So what are the nodes of Ranvier and what is their role? Nodes of Ranvier are the gaps between the myelinated sheaths, and their role is essentially to provide a place to jump. The signal jumps over them and transmits to the next one and the next one, and that's saltatory conduction, and it speeds up the transmission process. And explain saltatory conduction. That's what I just did. And then where does the name saltatory conduction originate? It's actually Latin, like most things, and it comes from saltar, which means to leap. And that is the end of our presentation.